Awesome. Good morning. Good morning, good morning, good morning. It's snowing outside. That means it's brighter in the morning, which has got to be a good thing, right? And it looks like we've got snow at least till Wednesday. Welcome. You got a big weekend, so I hope things went well. I hope that everything is going well, and I'm excited to spend time today talking with you about, we're moving into kind of our chapter about mobility. We're leaving behind, let me just erase this. We're leaving behind in the lectures the sensory systems that we've spent the last 3 different kind of shared times with. But you're going to pick that up this week in your labs with Shayla. And so our last time together, we were talking about mechanoreception. And there's kind of one slide today, or one example, one case study today, where we're going to be talking about spiders and how they move through the air, that has to do with some of the organs, sense organs, mechanoreceptors that the spiders have, that they use to help themselves move. What are we talking about this week? Well, we're going to be talking about, start off with right from the start. Let's talk about the ability to be model or mobile, synonyms there, or sessile, and why evolutionary pressures might have selected for one lineage or one strategy versus the other. And then we'll talk about, in the specifics of today's example, we're going to spend a lot of time talking about arthropods. Insects predominantly, but not exclusively. So we've got ants, spiders, feather beetles, moths, dipterans, and dragonflies. Oh my. And my hope is, in terms of what are the learning outcomes that this identifies within your class, is that you can appraise, synthesize, and interpret a range of invertebrate examples of mobile or sessile lifestyles, with the specifics of insect flight and the muscles and the systems that they use to do that you can bump into examples from the literature and in the world that you can interpret. And then to think about making a bridge all the way back to the marine systems at the start of the course when we were talking about plankton. Plankton's going to come up again today, but we're going to talk about aerial plankton, and we're going to talk about the role of inertia versus drag, and low Reynolds environments versus high Reynolds environments. So first of all, when we're thinking about mobility, the ability to be an animal that is mobile or an animal that is sessile. So mobile or model animals, you need sensory systems, right? And so we start thinking about, in more complicated models, we start thinking about cephalization. And then so that makes us think from an evolutionary perspective. In the Dunedal phylogeny, we start ruling out. So we're not thinking so much about the cnidarians, but they're mobile. And we're not thinking so much about the xenocoelomorphs, but they're also mobile. So it's a widespread strategy. And so when we think about, so we'll unpack kind of why, the pros and cons to why, and the variety of strategies that have come up over the next 2 classes. And when you think about sessile organisms that don't usually move, we think about them being attached to the substrate, and we think about sponges, and we think about corals, and we think about, not exclusively, but often colonial or marine lineages. And when we think about the mobility of those lineages, because from a genetics, kind of a population genetics perspective, or just an ecological, a metapopulation perspective, organisms need to have some mobility in their life history stage. And so for sessile adults, we often see mobility in their juvenile or early in their life history, juvenile or eggs. So what are some advantages to not being mobile? Like, why do we see it retained across a broad swath of the dun world? Largely, it has to do with conservation of energy. So thinking about the chapter where we talked about life history strategies, really we're thinking about kind of an energy budget and where an organism can allocate that energy towards. And in this case, the lack of energy spent on moving is energy that you can spend then on something like reproduction or growth. Disadvantages, it's really easy to be grazed or browsed, so you can be ripped off. You have to rely on your medium bringing you food, so flow becomes important. And this kind of thinking about the examples here, we've got sponges and corals and echinoderms, and we're thinking about organisms that basically set themselves up to be passive receptors. And thinking about our food chapters together, we're thinking about filter feeders, right? Largely. Animals the flow can be what you need, but it can also damage you. So high flow can damage you, and of course you're exposed to predation. And so there, the examples that we've got at the bottom, listed for the examples on the left, listed down at the bottom. So we've got sponges and different kinds of cnidarians, phoronid worms, and echinoderms. So you've talked with me, I've talked with you, we've spent a lot of time thinking about sponges. And so I'm confident that you know a lot about sponges. Then if you were to kind of categorize this for friends who don't have the privilege or the responsibility of taking and being a zoologist or being a zoology or marine and freshwater or wildlife biology major or an ecology major, if you were to simplify sponges, often you would start with, well, they don't move. They sit at the bottom and they don't move. They release their gametes into the water, and then a young sponge becomes sessile, and you might talk about different kind of feeding strategies. But often it'll start, and if you start unpacking feeding strategies, it starts from the perspective of sponges. What do they do? What do they don't do? Well, they don't move that much. And when we think about bilaterian organisms that are sessile, we see a reduction in cephalization. So when we think about the bilateria, kind of from the xenos, like our part of the phylogenetic neighborhood, but things that move through the air and that have a concentration of a nervous system up at the front end of the body, we see a reduction, a secondary reduction in that. And that the feeding apparatus looks more like lineages that have reduced or certainly are not bilaterally symmetrical. So we see the crown of the lophophores, or we see the crown of the echinoderms, the sessile sea lilies, like you just pictured there on the bottom. So cnidarian polyps, lophophorate, or crinoids. So the directionality of that resource is a thing. And what do I mean by directionality? When I'm thinking about flow, upstream, downstream, into the current, away from the current. This becomes really important in terms of eating and in terms of because you're not moving. So the saballid worms, that's kind of one example of a bilaterally symmetrical. This is an annelid, but it lives a very sessile life. So it lives in a long tube constructed in the mud or the sand. There's a couple of beautiful ones, these kind of like purple pom-poms, that have been pulled out, and you see this long curly Q and that on the top. That's lives in these long tubes, and they are, as we talked about earlier, they have a free living larval phase that's used for dispersal, but they are attached to the substrate, so we would technically consider them sessile, but they can use muscular contractions to kind of quickly retract into their tube. So there is some mobility in the same way that there is with a coral polyp, that they can also retract into their tube, right? Or a bryozoan, it can retract into its tube. And so here is one example, in and out, and here's the base. At the base of this tentacular crown, this feeding array, we see a mucus string that feeds into a mouth. Other kinds of annelid worms. This is like annelid worms. If you think about the kind of the mobile terrestrial example of the oligochaetes, or the parasitic examples of the horindimorphs and the leeches, or the fast paddlers of the homopterus, the polychaetes living up in the pelagic area, there are 1.5 zillion. That's a precise number. 1.5 zillion annelid lineages that are functionally sessile, that are deposit or filter feeders, like this turbellid worm that lives very much like the last example. So it's attached into the literature. It lives in there's a whole picture, includes kind of a whole bunch of them with their feeding tentacles spread out over the substrate, but they are creeping. They have body parts that are mobile, but the animal itself is sessile. So I think one of the things already that we're starting to unpack, or to shake the tree, when we shake the tree of Aristotelian 0 or 1, or a binary system, is that the difference between sessile and mobile is not binary. so little of biology is. So much of biology is it depends. Well, are you talking about the movement of the whole organism, or are you talking about having no movement of all of any of the appendages or the body parts? So in this case, if it's the whole organism, it's sessile. And certainly it's not if we're talking about the appendages. And if we become more familiar with the biology of lineage, that things we know, things that are certain to us, that sponges don't move, corals don't move. Well, in fact, as we learn more and more about different parts of the world that we haven't explored very frequently, like the bottom, deep parts of the ocean, as you explored in your Invert R experiments, but also certainly this sponge video on the bottom. This is a sponge that was named Belinda, that was filmed off the coast of UBC, so off the coast of British Columbia, but specifically nearby to the campus of the University of British Columbia. And when they filmed it seasonally, what they saw was that we were asking questions with regard to mobility in the incorrect time scale, that over seasons, these things do move. And the coral on top, this is a free-living coral that can get up and move around. And so our understanding of mobility is sometimes, as with so many other examples that we unpacked in detail in the sensory chapters, we're approaching the world, the invert world, with a whole bunch of our biases filtering our understanding of the world. Our vertebrate biases. These are naturally come by. Like they have served us well in evolutionary time, but they often don't help us when we're trying to be empathetic towards the vast diversity of the planet that is an invertebrate and that shares their world with us. So in this case, two specific cases from recent literature in the past 16 months that have shown that specific cases of sponges that are mobile and corals that are mobile. That's kind of amazing to me. And I also love that the researchers called this one Belinda, because that's amusing to me. So there's our first appearance of Caramel, and I'm looking down. I don't see any questions on the go in the chat. So this is kind of the base couple of pages of this chapter where we talk about pros and cons of being mobile or being sessile. And I think the takeaway message from this is that it's not a binary. There's a continuum, and we're going to spend the rest of our time on the far end of the mobility continuum with lineages that move a lot. And we're going to start out with lineages that move in the air. And so we're talking about mobile organisms, and when we talk about mobile organisms today, it'll be flying, but over the next couple of days, we're going to be talking about walking and running and crawling and swimming even. So the advantages for mobile organisms include an efficient strategy for acquiring food, a diversity of food. You're not restricted to flow. You become the flow. That makes me think of I'm the one who knocks in Breaking Bad. But you also can avoid if your environment becomes slightly toxic. You can move around. So from a reproductive perspective, you're not just restricted to those that are in the same kind of flow of the gametes that you've passively released into the medium in which you live. And you can relocate if your environment becomes too toxic or too hostile. So disadvantages are linked kind of inversely to the advantages that we talked about. The sessile advantages to being sessile and not moving really. And that has to do here with it takes energy. And so there needs to be some kind of a return. So in your energy budget as an individual and as a taxon, you need more food. And typically, mates are not distributed densely around you. Your potential for mating really has to do with search. And that brings in the kind of the sensory chapters, like how do you interact with the environment? How do you signal to your potential mate that you are in fact a really good strategy for them to follow up? So the types of movement, we're going to be talking about the flying today, but walking, running, crawling, swimming, we will come to that on Wednesday. So from a physical perspective, when we're talking about flight, we need to, and a lot of this will be reviewed because I'm presuming that you talked about this in various parts of your training way back into high school, but high school physics, high school biology, your vert course, you've talked about flying, and with the limited number of lineages that we're biased to thinking about that do it, your birds and your dinosaurs and your bats. But as Calvin and Hobbes once learned, bats aren't bugs. Bats did the flying, or bugs did the flying first, and everyone else had just kind of followed along millions of years later. But any of the lineages, from an evolutionary perspective, that do fly are exposed to these kind of forces. So drag, it's in direction to the fluid or the environment that they're moving through. Lift. This is perpendicular to the drag force. So the angle of wings and the angle that you creates a resistance that results in an upward force that takes you up into the air. And then the thrust vector is produced by powered flight. And it's mostly for the insects produced at the distal ends of the wings, and that's what keeps the animal moving forward. So as I mentioned, the first flying animals were hexapods. They were insects and their relatives. Then the wings that they've done it with are not appendages. So it's automatically its flight has taken upon various different lineages in the animal kingdom on our planet. And the very first lineage to do it, say 90 million years before the vertebrates did it, have done it in a very different way. So we don't use the appendages here. What we see, these wings, this is not a loss or an alteration of legs. Legs are for moving, legs are for walking, legs are for respiring. Legs are never for flying in a hexapod or an insect. How do you get your wings? Well, your lateral cuticle, which originally, in the same kind of strategy that might have been followed up with the birds, they might have been used as stabilizers or sails associated with things that glided or fell out of trees and didn't wanted to kind of move away from that object that they were gliding from. But you can see here this perspective. This is a little lateral outgrowth, a lateral outgrowth of the cuticle, that through evolutionary time, the spaces in between folded around those trachea, the trachea that are respiring and directly connected to the air in the external environment, get crunched together. So they're basically sandwiched in between these tightly in between these epidermal cells. And then through evolutionary time, that then gets reduced even more and more until it's basically just a membrane supported with these veins, and these veins are tracheal veins. And so this is how the rigidity of the wing is composed of the wing veins that are bringing out material from the trachea into the external environment, but also hemolymph. And so there's a pumping action that keeps the wings rigid, or as rigid as they need to be, but also can reduce that if you're going to be folding them up and hiding them. And folding them up and hiding them. So before we get to folding them up and hiding them, just a call to the fact that there are... Now, I seem to have put my hand up because I put my hand... Smith didn't. Oh my God. Well, we'll just leave it there. Or we'll change it. See if it reads the thumbs up. So the problem, or the what does flight do? Flight open is going to open up a lot of different niche space for you, from instead of being restricted to kind of a linear environment on the terrestrial surface. You can now, there's a whole dimension that you've added. Now, one of the ways that you can then further twist that advantage, if you're a flying insect, is your ability to hide those wings so that you can return to the terrestrial environment and then have the best of both worlds. You can move around, you can take advantage of all those advantages we listed for moving and that are particularly accentuated for those things that fly. And then you can hide them. And one of the best organisms at doing that amongst the beetles are a lineage that we've shared time with a number of times, and that is the these guys. These are staphylinids. So this is a rove beetle. You've seen a lot of different pictures of rove beetles that I've shown you. And I didn't film this video. This is a colleague, Adrian Smith, who does some incredible, he has a YouTube channel. You can watch his high-resolution, high-speed videos of insect mobility. Lots of different flying ones. But in this case, we're looking at the packing up of the wings that the staphylinid does. And it does it under a very reduced elytral space. And that permits it basically to move in the leaf litter or into the subterranean environment. And then when they're disturbed, they can come to the surface, pop the wings out from underneath the elytra, and fly away. I love that video. You could watch it for the whole class. I was considering just putting that video on and discussing things. So this evolution of flight and the folding of the wings gave hexapods a distinct advantage, that they could disperse, they could escape from predators, and then rapid access to food and to optimum habitats. Now, how they go about doing it has changed. Flight has likely evolved only one time within the insect lineage. That's also incredible when you think about the diversity of lineages, that this is not convergent, that this is likely lineages where we don't see flight, they have secondarily lost it. That's kind of speaks to how evolutionarily successful this was as a strategy. But from the lineages that first did it versus all of the lineages that now do it, we've seen different strategies in terms of how the muscles power the flight. So for the odonates, like this dragonfly that's taking off here, we see synchronous flight muscles. So there's each contraction is synced with an action potential that initiates it from the central nervous system. And then for the Paleoptera, so the Ephemeroptera and the odonates writ large, the wings can't be folded, and the wing muscles connect directly to the wing, and they insert at the base. And that's what this diagram is showing here. So we've got elevator and depressor muscles that are basically pulling the wing bases up and down. So the analogy that's made at the bottom is that this is kind of like the muscled rower sitting in the boat and then rowing that odonate through the air. Most lineages, that's not how it's accomplished. Most lineages accomplish flight via indirect flight muscles. So instead of the muscles being connected to the wings directly, they're connected to the thorax. And the thorax kind of pops up, so there's a reinforced cuticle on the top, and it's pulled down. Pop, like the lid on that spaghetti lid. So it pops in and out, and as it does that, it flaps the wings. And in addition, the innervation of the flight muscles is different. So they have asynchronous flight muscles that are not synced. There's many oscillating contractions. And this is seen in all of the other contemporary insect groups. So this is the mechanism that evolved one time, and then from then on, the same. And this is kind of, I have a video here in a second. So we've got the colors across these dorsoventral muscles that are contracted, and then we've got muscles running longitudinally across the length of the body that can change the flex of that cuticle. So in the upstroke, we see the dorsal ventral muscles contracted, that flaps the wings up, and then the downstroke, the tergite, that cuticle on the anterior surface, is contracted and then popped out. And so then we see the muscles coming down. And essentially, if you were to do a very coarse animation of it, that's what you would see. So this is indirect flight. And in addition to these large muscles, there's, of course, when you start thinking about it, as your brain has already kind of skipped ahead to, thinking about all the fine maneuvering that can be done by most insect lineages that fly, like the true flies, there's a whole package of other muscles. So here we see power muscles in kind of yellow and red, great big things that are connected, as you can see, to the cuticle. But as they zoom in this tomographic video, you can see all the other colors, yellow, green to blue. All of those are steering muscles. So a classic entomological text describes this as an absurd design, but one that ends up being, despite the apparent absurdity of the design, it's remarkably efficient and powerful. So muscles pulling on the thorax to move the wings, not muscles pulling the wings for the majority of insect lineages. When you think about the wings themselves, these little bits of kind of the lateral cuticle that you're going to use to flap around, again with the advantage, the technological advantage that you have being a biologist in this era, is that you have really, really relatively comparatively easy access to a whole suite of video tools, including high-resolution, high-speed cameras that researchers are using to look at and to learn more about how the kinematics of wing movements, how the wings flex and twist during flight. Because one of the first things that we learned as soon as we could reduce the speed of this was that the wings themselves, which you had maybe in very simple animation pictured as being like, again with the oar analogy, I suppose, thinking about them as being very rigid, they're not. They flex and twist quite a bit. And so we've got a video of a fly kind of moving from left to right, and then the diagram of this moth and the moth wing, you can see them kind of almost like sculling in the air. And that's actually a very common strategy. From the grasshopper to the moth to the bumblebee here, you can see there's the flapping. There is the movement that's provided by the muscles pulling on the cuticle that is flapping the wings. But there's also the smaller muscles that create a power and a recovery stroke. And we've talked about power and recovery strokes a bunch of times, but often it's when we're thinking about small things living in marine systems that use cilia, for example, that power and recovery, the kind of coordination of that, the nervous system. But there is a power and a recovery stroke to insect wings as they are moved through the air. And so so the wing here is this kind of hatchet or little flag. And you can see as you trace it around in this angle here, that hatchet gets moved, the head of that hatchet gets moved. And so there's a power stroke down that provides the thrust that moves them up into the air. And then there's a recovery stroke as it swings around to have a more efficient capacity to place the wing to have its next power stroke. So there's a twisting and a lot of deformation that happens in the wing. Now, when we think about the movement of these wings, either the flapping and the twisting that happens while the flapping has happened, because they're not disparate. As the flap happens, the twist is happening with the flap. We would measure that by hertz, and so we talk about wing beat frequency. So what I'd like you to do is to find your annotation tools and to give me some sketch on this graph. So we've got body size on the x-axis and the wing beat frequency on the right. And knowing you know about scale relationships back to the life history chapters, what would you expect for how size affects wing beat frequency? All right. Okay. Thank you. All right, let me put that annotation to the side for a second. And we'll show you what actually happens. And this is kind of scale independent. And by that I mean from the very, very small to the very, very large insects. So here across 3 orders, the neuropterans, odonates, and the butterflies and the moths, the relationship that we see there, and this has to do with muscles and how muscle strength is required, more muscles being required for greater wing beat frequency. We see this positive relationship between body size and wing beat frequency across all of these lineages. Okie doke. I don't see anything in the chat. Garamel's reminder is there. But let's think about frequency. Let's think about air. And let's think about, I mentioned that we'd be thinking about plankton again. So the aerial plankton, and this is when we mentioned kind of the evolutionary success, so like how many lineages, how many species of insect, how widespread mobility via flight is amongst the insects, this scales ecologically. And there's a couple of different studies that come out in the last several years that have tried to capture, to quantify the number of insects that are above you in any particular flight season, like this summer. So this is an example from the UK, where they quantified and they used nets to kind of do a subsection and then scaled up to say, if you were looking at basically a 1 kilometer vertical tower above you, there are millions upon millions of insects in that. So this is small insects, big insects being moved around. They are actively moving themselves around, but then there are many, many small things, much smaller than the moths or the ladybird beetles that we're watching here. And we always pictured or imagined or explained to ourselves that those small things, they are so small insects are very, very small, like down to the size of overlap with protozoans. There are amoeboids that are as big as some of the insects. And we always imagine that, but they have wings, and that these small insects experience the world and the air in the same way that planktonic organisms experience water, i.e. they don't move actively. And I'll call your attention or kind of remind you, think back to the Reynolds discussions that we had in our shared planktonic time together. So what kind of an insect is as small as an amoeboid? Well, there's a beetle, the featherwing beetle, Paratiposa placentis, which is this left-hand side, clearly, of the SEM. And then we've got the amoeboid at the other side. Now, small insects they have special challenges associated with flight that have to do with the flight mechanics at low Reynolds numbers. So they're beneath that kind of critical 1 millimeter, and they need to produce forces that support their body weight in conditions of high viscous drag, a high viscosity, and historically we presumed that they were air plankton and not capable of powered flight, and that has a lot to do with if they were going to be. How could this be possible when we see that positive relationship that we previously kind of elucidated together between the frequency, the wing beat frequency, and the muscular mass, and so body size, that positive relationship between body size and wing beat frequency. But as we begin to approach these questions with the technology, contemporary technology, you can see this Paratiposa platensis, it actually flies quite well. Actively. This is not just being carried around by the wind passively as a very small thing. And so they need to, how are they doing it? How are small, very small insects producing forces that can support their body weight in conditions of a high viscous drag? And there's a couple of different adaptations. One of them has to do with the structure of the wing, and another has to do with how they move their wing. So the diagram at the top, the diagram at the bottom and the video at the top are kind of showing the same thing. And one of them is they have one of the most accentuated power and recovery strokes that we've seen as high resolution and high speed cameras have recovered more and more insect lineages. So this is a figure 8 or an infinity that it's basically kind of looping back and forth. And so that power and recovery, almost like you're treading water, okay? And so the power, and think about this from the perspective, power, when we talked about muscles before, and this is back into 2700, that the power of a muscle varies in relation to its cross-sectional area, right? So that's a function of the square root of its volume, and that's then determined by the weight, or we measure it by the weight of the animal. And so as animals get bigger, their comparative strength diminishes because of this. So their weight and their muscles are going at linear and at cubic alternate ends of that equation. And so smaller insects have a bit of an advantage here because their weight, they're comparatively much lighter, while still benefiting from comparatively strong muscles. So instead of thinking of that last plot as an advantage, the positive increase, it's kind of a consequence of the biophysics of the square and the cube. So at low Reynolds number, they need to support, with this figure 8, they need to support their comparatively small body mass. And they're doing it partially with this sculling action in their bronze medallion of flight. They are still working they're a very small thing, so the air is more viscous to them. But how else are they supporting it? How are they using their comparatively stronger but still much smaller set of muscles to do that? How can they make that figure 8? And a lot of it has to do with the extreme reduction in the wings. So the Tylidae, this is the family of beetles that includes the beetle, the is in the title, the placentis beetle. And so the wing is hyper reduced, and so the veins are essentially reduced to teeth, or to these little thorns. And then we have thorns on thorns. So a very membranous and spiky wing that reduces the muscular power that's required to move it, but still maintains its aerodynamic properties at this very small scale. So a wing that's more like a net, or than an actual kind of like a continuous bit of your cuticle, and a figure-8 power and recovery stroke. And then you too can be an actively powered bit of the aerial plankton. There's other ways of, I mentioned we weren't going to be talking exclusively about flying things, but also about falling things and gliding things. And so we're going to bring up an ant example here. This is a tropical species that lives across Central America, certainly. And this is a picture taken of Cephalotes auratus falling off of a tree intentionally in Panama. This is a video where the auratus is taken out in front of the camera. This is an early digital footage. So this picture, this was about 20 years ago that this study came out. And at the time, so they're using the kind of the best cameras at the time. So they take the ant out to model what they had been observing as they worked in the canopy of this rainforest, which was ants seemed to quite intentionally throw themselves off the tree and into the space. And when they watched closely at what was happening, the ant, as you can see there, they don't just fall down. There's a directionality. They fall back using a very steep glide trajectory back to the trunk of the tree. And they're started to conduct more standardized experiments and to suspect that these ants were using visual cues. If you go back to the picture, you can see, it's hard to see it in this one here, but there's actually quite a few. There's the eye right there. So big compound eye out on the side of its head, and many ommatidia. So this is a really an organism that sees the world really well. And what their experiments showed was, when they modeled it, is that nearly all of them, and they're not passively gliding, but they're using some exerting control over their own bodies to the aerodynamic forces that they're experiencing as they're falling, to move themselves back to the trunk that they've escaped. So essentially they've seen you, the big lumbering primate, in the canopy, and they've jumped off the tree to glide back down to the trunk. So they've escaped you, but they haven't left the tree, which is, these are arboreal ants. This is their home. So they can vary the riding phase, the duration, the glide, the path, and if they bounce off, they try it again. And so we see different lineages, like the Cephalotes. This is the example we've been talking about, but there are other arboreal ants that can take their legs and spread them out and parachute down. And that's kind of incredible. And when we look at lineages of ants that display these kinds of activities, what we tend to see is that the ends of their legs are not big, beefy Arnold Schwarzenegger forearms. They're quite flattened, and those flattened tarsi likely allow them to use their legs to move them, to glide them actively back into the tree trunk. And we see this, so there's two examples, the Chromatogaster and the Cephalotes, two genera, but it's seen across multiple lineages that take advantage of the arboreal, the canopy environment. And it's likely not something that has evolved only once. It's come up several times. So this a strategy to get out of danger. This is behind the flying ants, or the raining ants, or the gliding ants. Now, other arthropod flying examples, we would be remiss if we didn't spend some time with Charlotte's Web and thinking about spiders ballooning. So the ultimate chapter of spoiler alert, if you haven't finished A.B. White's classic Charlotte's Web, you see all the babies kind of going up to the end of the fence post and leaving Wilbur behind and moving away, sending out these tiny, we now know, nanoscale fibers that they use exclusively for aerial dispersal. So in the air current, as soon as they the size of that fiber is again, just like as we were talking about the importance of the Reynolds kind of calculations with the beetle. For this spider, though, that's releasing those nanoscale fibers, the viscous force of the air is much more dominant than the inertial force, than the wind. So from the viewpoint, the quote from the paper that I love, or the science outreach of the authors of this paper, is that from the viewpoint of the spider silk, the air is much more like honey. So they just let it out, and then whoop, they get pulled away. Now, the link to the mechanoreception that I alluded to earlier is with the spiders. How do they know when to set it up? There was a seasonality that was inferred, that it's the fall and the spiders have to go away. Well, yes, in Charlotte's Web. But what we also now know to be true is that there are, and I mentioned these kind of mechanosensory hairs on the very hairy legs of spiders called Trichobothria. And the Trichobothria are receptive, they're mechanoreceptors, but they're receptive to atmospheric electric circuits. So the electricity that's in the air, as well as to airflow. So electric fields and airflows stimulate distinctive displacements of that Trichobothrian's hair, and that helps provide the signal that they need to move off, and they can start ballooning. So they kind of stand up. This is the behavior that precedes the ballooning, is that they stand up and they go up on tiptoes because those hairs have been triggered, and then they get pulled away. All right. Caramel's peering in on us, and in the last couple of minutes, we're going to go back to the moth and the flighted insects world. And it's, I think, important to think about moths because they're kind of amazing, but there are some moths, particularly sphingids, sphinx moths, that have great big muscles and power, very powerful and active flight. And they have some amazing adaptations that allow them to do that. So these are moths that can, as this one is showing here with its long proboscis kind of reaching out, they're hummingbirds, right? These are, they're living in the same kind of niche as hummingbirds do. So hovering is important for their feeding, and they can pump their abdomen up and down, and you can kind of see it if you watch the video very carefully, that they're fine-tuning the forces that keep them airborne, causing the center of mass to change. But even in addition to this, they have some, so the pumping there is shown by the displacement, so the direction when the wings are going down, the abdomen's going up, and when the wings are going up, the abdomen's going down. So there's kind of like a teeter-tottering that's happening that allow them to keep them airborne and keep them in a very stable position from which they can feed and not break off their proboscis. Now, one particular lineage, this death's head hawk moth. So there's the big gorgeous caterpillar, and here is the adult, and you can see kind of the scales on the thorax that array to look like a little bit like a skull. So that's the root of the common name, but the death's head hawk moth. This is a look at its thorax underneath the wings, and we're looking at these two, this hole right here. These are spiracles, so the opening of the tracheal system to the external environment. Because the spiracles that are placed on this sphingid's body are opening and closing in synchronization in different parts of the body with different parts of the wing stroke. So the spiracle is closed when there's a downstroke under the contraction. And I'll show you I'll break out these texts with some diagrams here in a second. But it's creating essentially a unidirectional ventilation. So there's an inflow. They have anterior on the front end of the body as the insect's kind of moving through the air. So on the anterior end, the thoracic spiracles are, let's do the diagram so I can picture it. So the inspiration. So the spiracle is closed, and so there's the spiracle under contraction of the big flice muscles that's contracted there. And so we see a contraction and a wing beat. And then the air sacs in the thorax, that's this, expand, and air is sucked in at the front. And so we see the opposite when the other longitudinal muscles contract and the wing beat goes up. And we see basically that air is sucked in at the front and pushed out at the back. And that's providing a jet propulsion. There is a little bit of the expulsion from this tiny bit of and kind of more posterior spiracle is providing two things. It's providing a really analogous thrust force to what jet engines use, but it's also allowing a great big gasp of air to oxygenate the muscles, the big giant flat muscles that are doing all of this work. So they're acting essentially as a big, like the bellows for your fire, or a big colossal suction pump. And they're sucking air in at the front of the insect before pistoning it out at the back end. That's kind of amazing to me. And we've talked about halteres before with mechanoreception and chemoreception. So we've got little bits of hairs, but we're going to talk about it again with flight, because the synchronization of these, so these are the little halteres. So these reduced and the true flies, the dipterans, the hind wings are basically these mechanosensory organs that detect the body rotations and maintain stability during flight, allowing them to zip and zag and dip and dive. And there's kind of two orders. Let me go back to that, because there's two orders of insects, or two groups. This is within the dipterans. So they're all still flies, true flies. But there are calypterate that use the halteres to stabilize their bodies in walking and flight, and then non-calypterate dipterans that don't do that, that it's only during flight. So here is the big kind of drumstick writ large. So this is our drumstick, but we're looking at these very tiny little reduced wings. And we talked about the fact that they're kind of engaging and disengaging, that there's a clutch that permits them to act as decouples and uncouples, or couples and decouples with the wing, using these thoracic kind of cuticular extensions. So on the top, this is a fly. So we've got left haltere, right haltere, and the wings. And you can see this is kind of their displacement over time. And you can see that when they're walking, this is the non-calypterate. So walk, 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 walk. And then as soon as they start to fly, there's a little bit of a lag, but then the halteres start to fire and start to synchronize with the larger wings, the forewings. In the bottom, you can see there's like a back and forth that the left and right haltere are doing. So they're coordinated. They oscillate in non-flight. So as the fly is walking, so the walk, that they're coordinated. And then you can see a much more rapid synchronization when they take off. Kind of an amazing strategy, and the subtleties of this strategy, of the functionality of the haltere, we're kind of still trying to figure out across different lineages, and we're still trying to take advantage of them from an engineering perspective or a biomimetic perspective. What can we learn about more energy efficient flight from looking at insect designs that have been successful for millions and millions of years? For example, from the dragonflies. So there's a huge amount of muscle that goes into that, right? And remember, they are the left-hand model, the odonates. So this is direct flight. So the wings are being pulled on by the muscles instead of this, which is more of a fly example. So very large flight muscles here in the odonates. 60% of their body mass is dedicated to flight muscle. And that's the highest ratio found in any insect order. And they can fly very fast. And they are, if you think back to our feeding chapters together, when we talked about predation, they are amongst, if not the king of the world, king and queen of the world in terms of predatory efficiency. Sometimes 90% of the time they will get. So this puts the lions and tigers and bears to shame. So direct flight using 60%, and that number kind of stuck in my head as I was preparing the slide. And so I thought, well, what does it look like? What does 60% of your body mass dedicated to flight look like in a human? And it basically looks like the Hulk. This is what you would look like if 60% of your mass was dedicated to the muscles that power how we would fly. Now, outside of kind of the science fiction applications and inside of, so not too distantly related, so the science fiction and the engineering perspective, as we learn more and more about the subtleties with which dragonflies and odonates fly, what we've learned is that they can fly upside down and backwards as efficiently, as energetically efficiently as they fly forward. And that has to do with the fact that when they angle, when they take off, they're angling their bodies up. And they use their wings to pull back in the same way. So as you took off from the trunk of a tree, you're not having to fly up. You can fly backwards. And they're using their whole body kind of rotating around to do that. And so if we looked at where the tips, and I mentioned earlier This is, the colors here are in relation to where the most force. So a more red color is more force. And you can see, I mentioned that it was at the distal ends of the wings, the tips of the wings. This is where the force is coming from. So they're doing it as energetically efficiently as backwards and upside down as they do flying forward. This is intriguing to engineers all over the world from a human flight, assisted flight perspective, and potentially thinking about biomimetic solutions to aerial robots, if not planes or helicopters that we would fly in, but drones that could be parameterized to fly in a much more Odinate-like fashion. Fast, upside down, backwards. And this is kind of so thinking about drones, but then also thinking about science fiction and thinking about the Dune movies, the ships in science fiction in Frank Herbert's book back into the 60s, but also the Denis Villeneuve's contemporary adaptations with Timothy Chalamet Ding Dong and Zendaya is essentially an Odinate. But it extends beyond that because we've actually built Dragonfly drones in reality. This is the promotional video, actual footage of a Dragonfly drone that they have built, and that before the pandemic, this particular company was trying to market. So expect to see more flying insects in the drone world, more insects or drones where their design is derived from what we understand about flight from observing the insect world. All right. That's about the time that we've got. So I hope that this was enjoyable as we talk about the basic pros and cons of being mobile and sessile, and then understanding that it's not a binary, and then thinking about different examples, not just the ones that you might readily think about, the flies and the Odinates, but also the really tiny things, or the really improbable folding wing things, or the ballooning and using the electrical currents of the world to move around the world. On Wednesday, we're going to be talking about walking and swimming and crawling and running. In the lab this week, you're going to pick up the sensory systems and finish that off with Shayla. And what to be working on? Well, I hope that you will be working on your lab report, which is due next week. And then check out course link, but in relation to the lab that was missed last week, the deadline for your final video presentation has been moved to the 26th, which is the last day of the semester. So I'm going to sign off. Take care of yourself. Take care of those around you. And we'll meet up again on Wednesday. And if you've got questions, hang around for a second after I stop recording and stop sharing and ask away.